Hello everyone, and welcome to another edition of our MJS BioLinks Breathe and Learn. I'm Steve McKend. I'm probably the tiny talking head in your corner if I'm not pinned. I'm in business development here at BioLinks, and it's my pleasure to introduce our guest today. We're joined by Andrea Johnstone from EpiCypher, who will talk to us about the paradigm shift that's happening in chromatin research. The introduction of CUT&RUN and CUT&Tag is solving the problems that have been limiting the application of genomic mapping, such as poor sensitivity, low throughput, and high costs. So today we'll explore what next generation chromatin studies will look like with the help of these new techniques. And as usual, this will just be a quick 15-minute demonstration. And if you have any questions, feel free to throw them in the chat below, and we'll get to those immediately after the demo. And before we jump in, I was hoping we could make this a little more personal. So if you wouldn't mind changing your Zoom name to your real name, if you feel comfortable doing so, just go ahead and click on your little tile and rename yourself. And if you want to go one step further, you can even throw on your camera. So while you do that, I will run through the necessary webinar reminders. First of all, this session is being recorded and we will share it with you right afterwards. Second, make sure you pin Andrea on your screen to keep her tile in view as she shares her presentation. And out of courtesy to everyone on the call, I have muted all of you until after the Q&A section. So with that, I'll pass it off to Andrea. Enjoy the demo.
Awesome. Thanks so much, Steve. And thanks so much to the whole MJS BioLinks team for hosting this. I'm really excited about the format of this Breathe and Learn style webinar, and just to have a more informal conversation after I just give you, the highlights of some of these technologies we're developing to better understand how chromatin is regulated. I'm interested to hear your thoughts, your questions, your pain points, any of the above. So first, let's start with the basics. Why do we care about chromatin? If you're here, you probably already know that or appreciate that. But essentially, chromatin is, you can think of it as the gatekeeper in the cell between all the genes that exist in a cell at any given time, and which of those genes are turned on in response to genetic or environmental stimuli. And there's a diverse array of regulatory mechanisms that influence chromatin accessibility and turn genes either on or off. And so chromatin is very dynamic, but it primarily exists in a more open state associated with gene activation. And this is associated with specific modifications to the DNA and the histone tails, like histone H3, K4 methylation, for example. But chromatin can also be more compacted and condensed to repress and silence genes. And this is associated typically with DNA methylation and marks like H3K27 and K9 methylation. And upstream of these histone and DNA modifications is an even more diverse array of signaling pathways, including the enzymes that add, interpret, and remove these modifications, enzymes that recruit to the chromatin in order to shift things around, remodel them, and make them more accessible for transcription factors to ultimately bind and turn genes on or off. So it's really essential to have really robust technologies that are capable of mapping the localization of where these histone modifications and chromatin proteins are localized. Where are they found across the genome, and how are they changed during the course of normal development? And finally, how can they go wrong and contribute to disease, potentially leading to novel therapeutic strategies? And one of the most widely used technologies to do this is chromatin immunoprecipitation followed by next generation sequencing. And this has really been important to better understand chromatin regulation, but unfortunately has major limitations, including requiring large numbers of cells, really laborious, low throughput workflows, and very high sequencing costs in order to resolve only modest signal from noise. So your data quality at the end of the day is subpar, even after this labor-intensive, expensive workflow. Now, in contrast to ChIP, where you're essentially trying to immunoprecipitate your target amongst a mess of background fragmented chromatin, there's a novel technology called CUT&RUN, which utilizes this immunotethered enzyme, this protein AG, conjugated to micrococcal nuclease, which cleaves antibody-bound target chromatin and liberates it into solution. So now you've left all your background behind in the intact immobilized cells, and only your target DNA is released into solution. And this enables dramatic gains in sensitivity, so you can drive to dramatically reduced cell inputs. The workflows are very user-friendly, amenable to automation, and robust. And because of the gains in sensitivity, you can resolve signal from the background even at dramatically reduced sequencing depth, approximately 10-fold reduced sequencing depth. And so here's a look at what some of the cut-and-run data looks like in a head-to-head comparison of the historical approach, ChIP-seq. And so these are two tracks for a mark of active gene promoters, H3K4 trimethylation, and a more broadly dispersed mark of repressed genes and energetic DNA, H3K27 trimethylation. And what you can see is that the signal enrichment in ChIP versus cut-and-run looks largely the same. However, let me draw your attention to the number of NGS sequencing reads that were used to obtain this data. You can see cut-and-run gives you pretty much the same signal at 1/10 of the amount of sequencing reads, just 4 million compared to almost 40 million reads. And even at that reduced sequencing depth, the data quality in cut-and-run is actually higher compared to ChIP. So if we look at data quality using an unbiased measure of signal to noise, this genome-wide analysis of the fraction of reads that you see in the peaks, you can actually see that this is higher in the cut-and-run data compared to the ChIP, even at that reduced sequencing depth. And so this enables you to drive to much lower cell inputs, as I said, and that's shown here where we did cut-and-run for a lysine acetyl reader protein, BRD4. And what you can see is the signal in red at annotated transcription start sites is largely retained as you drop the cells from 500,000 cells, which is the recommended starting point, down to just 5,000 cells. You can see that the signal is pretty much the same with no change to the standard protocol. And so another good piece of news here is that cut-and-run, as you can tell from some of that data I showed, not only works for histone PTMs, including methylation and these labile acetyl marks, but it also works for chromatin-bound proteins like transcription factors, these writer and reader enzymes, and interestingly, even remodeling enzymes, which are very difficult to profile by ChIP because they interact very transiently with chromatin. And these are really high-value drug discovery targets. So the protocol is robust. It works for diverse targets. It also works for diverse sample inputs. I'm showing you here, these are suspension cells. It works for adherent cells, nuclei, lightly cross-linked or native cells. But one thing we've actually been surprised to see, well, it works for tissue, that's been reported in the literature, but there's an immense need in the field to obtain epigenomic maps for clinical samples. And frequently these samples are flash frozen, stored somewhere in a freezer, and people thought that we couldn't access them for ChIP or any type of epigenomic mapping approach. But we've been doing this in collaboration with Shahzad Sheikh at UNC. And what we did is took a single intestinal biopsy. These are small chunks of tissue that were flash frozen. And we were able to simultaneously map transcription factors and histone modifications of pathologic relevance. And we were very surprised to see that the data quality is actually similar to fresh cells. And so this is an ongoing collaboration where we're trying to identify epigenomic biomarkers of Crohn's disease that might inform future treatment strategies. Okay, so the sister technology of cut-and-run is called CUT&Tag, and this has gained an enormous amount of interest in the field, in particular because it uses a different enzyme compared to cut-and-run. So in cut-and-run, we use PAG tethered to micrococcal nuclease to simply clip your targets into solution. In CUT&Tag, instead of nuclease, you have transposase, TN5. And what that enables you to do is directly tagment your sequencing library adapters to target chromatin. And this enables you to do epigenomic mapping in a single tube. You don't have to do library prep and add all those extra steps associated with that. You just PCR amplify your libraries. So you can go from cells to libraries in one tube. Sounds fantastic, right? And in fact, it is. It enables you to drive towards dramatically reduced cell numbers. So here I'm showing data that you can go down to 1,000 cells in the standard workflow. There's also publications that have integrated CUT&Tag with combinatorial barcoding or microfluidics to go to single cells. But one caveat of this approach, you do have to wash more harshly compared to cut-and-run. So it's really just ideal for stable histone PTMs and some key transcription factors. So I'm showing you data here from a variety of histone modifications where we generate the expected profiles. CTCF has a little bit longer dwell time compared to most TFs, so it does work for CTCF. But we actually tested 18 different transcription factor antibodies that work well in cut-and-run, and only 3 of them work in CUT&Tag because of those harsher washes are stripping those lightly bound or transient interactors away. So CUT&Tag is really transformational, robust, and streamlined, but it's really ideal for when you really need to go to low cells and you're primarily profiling PTMs. For other applications, we recommend CUT&RUN as the go-to assay. But regardless of which assay you're using, any epigenomic mapping assay has one major problem. You start with cells, you put those cells through a series of steps. Maybe you have some quality control checks along the way, but for the most part, it's a black box. You don't know exactly what's happening in all those steps. At the end of the day, you get data, but it's very hard to tell if your experiment worked, especially if you're knocking down your target or drugging your target and it's depleted from chromatin. How do you know that the experiment worked or that it failed and you just saw reduction in your target? So we really need to find controls for that. Another thing is we know there's a reproducibility crisis in the field. Asking an antibody to distinguish among highly related histone states, mono, di versus trimethylation or methylation at distinct sites is a very big ask. And we know that histone PTM antibodies are notoriously not specific. So we need to account for that in our mapping assay. And then at the end of the day, the data are really qualitative. How do you cross-compare samples? And so to address some of these questions, we turn to one of EpiCypher's core competencies, which is developing fully defined nucleosomes. These are very hard to manufacture, but we've been very fortunate to be funded by the National Institutes of Health here in the United States. And we've been able to generate a catalog of over 100 different flavors of nucleosomes. And we've published showing on how you can use these for antibody specificity, chromatin binding assays, epigenomics, PTM mass spec, and a variety of other things. And two of the tricks that we can do with these nucleosomes is engineer distinct or defined histone modifications wherever we want on the histone tails, and also wrap these nucleosomes with unique DNA barcodes. And this enables us to assemble these nucleosomes into rational panel sets that constitute the target in your assay, for example, H3K4 tri, versus related off targets. So for example, H3K4 di or monomethylation. And what we do is we add these into either CUT&RUN or CUT&Tag assay early in the workflow. They resemble the physiological target. The target in your mapping assay is nucleosomes, the chromatin. And so these defined controls are carried through alongside the sample chromatin, the entire workflow. And because they're DNA barcoded, you can then in the resulting sequencing data, ask which of these spiking controls did I recover? Was my antibody specific? We've done this huge study that is publicly available on chromatinantibodies.com to test histone PTM antibody specificity in ChIP-seq, actually, finding that over 70% of them fail. But using these spiking controls, you can actually find ones that are good. Another way you can use these spikings is to confirm that your assay worked. You should recover your target and no off targets. If something went wrong there, that enables you to try to troubleshoot and improve your assay moving forward. And then you can do quantitative normalization because you're spiking in now a defined standard. And so what we've been doing using these spiking controls is finding highly specific antibodies by testing them directly in CUT&RUN. So this is a heat map showing specific antibodies for widely studied PTM states. And we also use these spiking controls to drive to low cells. You know, as soon as you reduce cells, if the antibody's not kept busy enough, it might start to cross-react. But we've been developing antibodies where we're titrating down to very low cell inputs, making sure the signal is on target and specific at all of those, and making sure that in the sequencing tracks, we're still able to resolve good signal from background. And then finally, these spiking controls really enable us to go take epigenomic mapping into new frontiers. Can these be used for precision medicine approaches where we're trying to understand biomarkers that are associated with disease, or read out direct measures of patient responses, or inform new treatment strategies? And so this data that I'm showing here is using an inhibitor to this protein menin, which catalyzes K4 trimethyl. And this is a really popular drug target right now. Several companies, including Syndax, are developing menin inhibitors that are currently active in clinical trials. And what I'm showing you here right now is CUT&RUN data where we were essentially taking cells treated with the Syndax inhibitor that were mixed at defined ratios with DMSO controls. And what you can see is a linear reduction of the menin recruitment to chromatin, coincident with a reduction in K4 trimethyl, the downstream target of menin. And so we're starting to use this data to uncover which genes are impacted by menin inhibition. And this type of approach could be applied for other drugs as well. Okay, so if you want to get started, I really recommend starting with CUT&RUN as a go-to assay, especially if you're new to these type of approaches or you're coming from ChIP. We sell all the key reagents and open access protocols directly from our R&D team. But if you want a little more user-friendly solution, we have kits to take you from cells all the way to sequence libraries. And we have services right now that are in beta launch mode, so you can inquire if you're interested in that. But if you kind of know what you're doing, or you really have a need to go to lower cells, CUT&Tag is a great way to do that. Again, we sell all the individual reagents. We have our optimized protocols directly on the website. We're gearing up to launch kits early next year. And we have active ongoing R&D for really innovative applications of CUT&Tag, like single cell, spatial genomics, all kinds of fun stuff. Don't forget your controls. As I talked about using those spiking controls, we use those to validate lot specifically every antibody that we sell, or we can recommend other antibodies from other vendors if we don't have them. And we sell antibodies for both histone PTM and chromatin protein targets. And then we have always active R&D programs. We're starting to venture into DNA methylation assays, chromatin accessibility, multi-omics, where you're layering these different chromatin features on top of each other to get a more comprehensive view of what's going on, and even coming up with bioinformatic analysis solutions for people who maybe are not bioinformaticians themselves to be able to analyze and interpret this data. All right, so thank you. I just want to acknowledge our funding through the NIH, all our very talented scientists who are so much fun to work with. And again, CEHRC and MJS BioLinks for sponsoring this. And if you guys liked what you heard today, please reach out to MJS. I know they have some promotions going on right now for some of these products through the end of the year. So I'm sorry if I talk fast, but hopefully you caught all that. Thanks.
Thanks, Andrea. That is not an easy subject to squeeze into 15 minutes, but well done.
Thanks.
So we do have one question here from Pamela. Can the cut-and-run samples be tested prior to sequencing? So for example, by qPCR or some other method to validate or check samples for expected targets?
Yeah, that's a great question. We get that a lot. So people are used to doing this with ChIP. We don't recommend it because the fragments that you get in cut-and-run are a little bit shorter than what you're typically used to seeing in ChIP. So if you're designing primers to your target of interest, you have to get it exactly right. It is possible, some people do it. We don't recommend it just because since you're only sequencing 3 million reads, we recommend 5 million reads is what you should shoot for. The sequencing costs really aren't that expensive. So the best thing that we recommend is just doing a size distribution analysis, either like a tape station or bioanalyzer. If you see a peak that's enriched for mononucleosomes, you're good to go on sequencing. It's going to work. It has never not worked as long as that QC check looks good. And we recommend other QC checks throughout the protocol, like making sure your cells have good integrity, they're bound to the beads, looking at DNA yields, is your target a little bit higher than your negative control? So in our hands, those are always sufficient to tell us, hey, the experiment worked, let's go forward with sequencing, and we're already saving a lot of money compared to ChIP, so don't waste your time on PCR.
Awesome. Any other questions? Feel free to toss them in the chat. We'll give you a few minutes.
Well, people are thinking of them. I'm Jen, I work in the technical support team at EpiCypher and have some FAQs that we can kind of have Andrea answer if you're not ready to ask a question yet. So, something I get asked a lot is I've heard transcription factors don't really work well in cut-and-run, but yet you showed some in this presentation here. Was there any change to the protocol or special trick to get those to work?
No change. So the biggest key is finding an antibody that works. A lot of people like to go straight from whatever they did in ChIP and assume that the same thing's going to work in cut-and-run. And it's not always true, right? Antibodies are not universally capable. They're good at some things and bad at others. So the first thing we recommend is find an antibody that works in cut-and-run. You can start with your ChIP antibody, but we recommend either sourcing a few more, testing 4 or 5 antibodies side by side, and almost always you can find one that works. Another trick that can help, some targets are very labile. or they bind to labile modifications on the histone tails. So lysine acetylation is rapidly turned over by histone deacetylase enzymes. And what we found is that if you very lightly cross-link your samples, not a standard ChIP fixation, but a light cross-link, and we have protocols on our website for doing that, it stabilizes those marks enough so that your chromatin-bound protein might have a longer dwell time, and then you can profile it. So we've seen some targets that use those labile acetyl marks to anchor will benefit from a light cross-link.
So if you see some targets work well with cross-linking, should I just cross-link for all of my samples?
No, that's a good question. No, because cross-linking does reduce your yield in cut and run. So it's always ideal to start with native cells. That actually works for most targets. Even some histone acetyl targets, which I've just told you are labile, even a lot of those work fine under native conditions. There's just very specific circumstances where a light cross-linking might improve your signal, but you're always fighting the reduced yield of that.
Any questions from the crowd? I have more to go through. Okay. So we've talked about ChIP-seq a bit. Is the data analysis for cut and run similar to the data analysis for ChIP-seq?
Pretty much similar, yeah. If you know how to analyze data with ChIP, it's the similar sort of workflow. You're using FOTI2 to still align your reads to the reference genome. You can use the same peak calling program. We use MAX2 a lot. We have actually switched to using CySER as well, which is commonly used for ChIP. There is some consideration though, because you're not running an input sample in cut and run as you would for ChIP. Instead, we recommend running an IgG negative control. So any peak calling or things you're doing are using that as the baseline point of sort of your background signal. But other than that, it's very, very similar to ChIP. We do recommend looking at the spike-ins as well, and just to make sure that your experiment worked by checking the, did you recover the target nucleosomes that you spiked in to the workflow?
Cool. So with these spike-in nucleosomes, what samples can they be used for? Like what targets?
Yeah, so the current panel that we have is for lysine methylation targets. We're gearing up to release panels for lysine acetylation targets and ubiquitination targets. If you're studying transcription factor, we have a way of making spike-in nucleosomes for transcription factors as well. It basically relies on knowing the antibody that works, and then we can make a nucleosome where the epitope of that antibody is actually fused on to a histone tail, and we use that as an on versus off target control. And so we're starting to use those in our service projects now, and we'll probably launch some really popular studied targets hopefully next year.
So during your talk, you introduced two different technologies, cut and run and CUT&Tag. Why should I use one versus the other?
Yeah, a lot of people like to go straight to CUT&Tag. It sounds cool. It's one tube, very streamlined workflow. You don't have to do library prep. But like I showed in the presentation, it does work very well for histone PTMs, but because of those harsher washes, some transcription factors don't survive. So we recommend, especially if you're new, start with cut and run. It pretty much works in all cases, but I would only recommend going to CUT&Tag if you have a specific reason to do that. If you really want to get to low cell or single cell, or you want to do some innovative application like spatial epigenomics or something like that, the PAG TN5 can work for those types of applications. But as a starting point, cut and run is really what we defer to internally. It's what we use in all our service projects. We have it automated on in 96 well plates. And so there's nothing about cut and run that would prohibit you from sort of scaling and doing larger scale streamlined studies.
Okay, so for either of these assays, can I use either like tissues that I have flash frozen in the freezer or just tissues in general?
Yes, you can use tissues. It's always better to use fresh tissue if you can, if you have access to that. And if you can, take fresh tissue, disperse the tissue into just single cells. So downs or, break it up with detergents. We have recommendations for that. And then once you get to single cells, cryopreserve them or start cut and run right away. If you have samples that were flash frozen, like those clinical samples that I showed in the presentation, those can be used in cut and run as well. Our tech support team can probably offer suggestions for that. And we're hoping to publish the protocol that we've optimized in-house for that as well. But surprisingly, flash frozen tissues can work. It just takes some tips and tricks to get good quality nuclei from those samples.
Yeah. Anyone is planning to do that, feel free to reach out to us. We can help with that. So something I've heard from people quite a bit is having difficulty getting reproducible data. Do you find that an issue in your hands, that the data is not reproducible?
No, in fact, it's quite reproducible. Like Pearson correlations are typically above 0.95 or better. I think the key thing is in all our protocols and our manuals, we recommend quality control checks throughout the protocol. And one of the biggest things is starting with really high quality samples. Your assay is only as good as your samples, right? So the better your samples, you want cells that are intact or nuclei that are intact. The more lysis you have, the more DNA that's going to be out there and it's going to be sticky and kind of cause things to clump throughout the protocol that could negatively impact your reproducibility. And then the other thing is finding a good antibody. If your antibody is not really efficient, it's not really good at grabbing your target, that could contribute to poor reproducibility as well. So we can provide recommendations for antibodies. We do sell some and we always check them not only for specificity, but also for high efficiency. Or we can make recommendations for antibodies to targets that we don't sell.
Thanks, Jen. Do you have one more quick question? We're coming up on 2:30 here. This has been really intriguing to watch. If you have one more, we have time for one more.
So yeah, one that I get quite frequently is that people experience a lot of clumping in the Can beads. That's the solid support Andrea mentioned that the cells bind to. Do you know how to avoid this?
Yeah, that's probably, I'd say, the most common problem that people can encounter. And we definitely have ways of overcoming that. The first thing is don't freeze your Can beads. Unfortunately, that sometimes happens during shipping. And that's we're working on ways to better control that. But when you get your beads, if you haven't frozen them, maybe just give them a check and look before you even use them. Make sure that they're not clumped when you receive them. If they are, let us know or let MJS know, and we can address that. But on your end, don't freeze them. Put them at 4 degrees. And then throughout the protocol, we recommend a whole bunch of sort of important steps for pipetting, vortexing the samples, making sure you add detergent to kind of loosen things up to avoid clumps. And so just following those key steps in the protocol are tips and tricks that we've learned doing this to help to avoid the clumping. Oh, and then the other thing is quality samples. So if you have a lot of lysis in your samples, things are going to get sticky and that will cause clumps.
Yeah, that's what I was going to add to it. And in addition, this is one of our key QC checks. There's a few others, but this is major. Your cells have to bind to these beads. If they don't, you're not inputting any sample into the reaction. So for sure, if you're going to try this, definitely do that QC check. And if the cells bind, you're inputting them into the reaction, and you should have DNA by the end.
Yep.
Love it. Awesome. Back and forth, you two. Thanks so much for doing that. And I guess that'll conclude our session for today. Thank you everyone else for coming in. I hope you enjoyed it. If you have any more technical questions or looking for ordering information, give us a call or send us an email. We have an awesome tech team that would be happy to help. And as you can see on the screen, we are currently running a bundle promotion. So you can receive 15% off if you buy both a cut and run kit as well as a library prep kit together. And we'll also follow up with you soon to share this recording and get you that Tim Hortons gift card that I know you're probably waiting for. All right, everyone. Thanks so much.
Thanks, everyone.